The team asked me just to give you a quick walk through CELMoDs, and obviously it's great that we're all here together, and in particular Sagar as well, because I'm actually going to start first of all with some discussion around iberdomide. Next slide, please. Obviously, as we understand the issue of how CELMoDs work, I mean, I think of them broadly as kind of molecular glues, and obviously in that sense, they're so-called monodacs. But I do think it's important to understand the distinction with the immunomodulators. I mean, these, in my opinion anyway, are clearly not glorified IMiDs. They're distinct and different. And I think the important construct summarized in this particular diagram, which just helps us understand this engagement with the Cereblon E3 ligase complex, and the potencies of this molecule of these molecules rather, depends on this engagement, which results in this highly targeted degradation of Ikaros and Aiolos. And downstream of that, not only is there this direct effect on the myeloma cell, but also, of course, these profound effects on the immune system, which are obviously in the era of T-cell redirection and CAR T therapy, also particularly interesting. Next slide, please. So with this in mind, I just wanted to quickly move through some of the data, and first and foremost, acknowledge Sagar's work with iberdomide in the 001 study with 220. And what was seen here with the combination of iberdex was a solid response rate, but above all, excellent tolerability. I mean, really, iber is, and certainly Sagar can speak directly to this, but certainly in our experience with it at our center, it's been a remarkably well-tolerated drug, and essentially a very solid set of response rates in the studies that we've done so far, and durable responses, recognizing the limitations of just these essentially monotherapy studies. Next slide, please. So as we build combination strategies, I think herein lies some important clues. When iberdomide was combined with daratumumab, the response rates improved. There was the expected synergy with both bortezomib and carfilzomib. Interestingly, not an obvious difference, recognizing these are small numbers between the two proteasome inhibitors in terms of potency. We might have anticipated a stronger signal from carfilzomib, but that certainly wasn't immediately apparent on these early phase studies. And again, durable responses from the combinations, really magnifying the degree of clinical benefit. Next slide, please. So when we think about mezigdomide, just a few points to make here. This is from the paper earlier this year, in fact, just last month, from Dr. Short, who presented, I think, a very informative editorial on the construct around the CELMoDs, and in particular looking at the molecular characteristics of mezigdomide and how it works. I think that the most informative part of this diagram, to me at least, was panel C, where we understand this ability to close the Cereblon complex, and recognizing that with pomalidomide, which I think we all agree is a highly potent IMiD, nonetheless, the degree of closing of the Cereblon complex is around 20% in experimental models. And with iberdomide, it improves to around 50%. And then remarkably, with mezigdomide, it's actually 100%. And this may explain some of the potency differences that we see. Next slide, please. Quickly to move through the mezigdomide data, because everyone's familiar, but just to show you some patient cases, which I think are also much, so informative. Obviously, in the phase 1/2 experience, we were able to demonstrate a solid response rate at the recommended phase 2 dose of around 55%. When we took this out further into the expansion cohort, it was 41% overall, but what struck us was activity in extramedullary disease, which was impressive for just a doublet. And we'll come to some patient-specific examples in a moment. And then what was also very interesting, but just want to be careful because it's a small number, in the 30 patients who've been BCMA exposed, predominantly antibody-drug conjugate, but also bispecifics and a small number of CAR T patients, we were able to show a 50% response rate, which we thought was provocative. And again, a manageable safety profile. Hematologic toxicity dominates, neutropenia in particular, but really in terms of our significant non-hematologic toxicities, these were modest and manageable. Not trivial though, fatigue was there, certainly some occasional cases of dysgeusia and so forth, but again, broadly very manageable. And what I thought was very important was the infectious profile. In terms of the infection profile, during the same period that we enrolled to this study as was, for example, enrolling to some of the bispecific studies at the height of the pandemic, and we only had one COVID death of 178 patients. And I think that's an important observation. And this was an outpatient platform, obviously, which was another attractive aspect for our patients. Next slide, please. So in terms of the EMD work, which I really wanted to show just because Lauren and the team had asked me to be patient focused. This is one example from Nizar's practice. And Nizar showed this patient, I think probably several of you have seen this before. This is a patient with a significant hepatic extramedullary plasmacytoma and an outstanding response to therapy. And basically after 3 cycles of mezigdomide had an excellent response. And this is a feature we have seen. Next slide, please. This is my patient, and actually, if you look at the treatment start at study entry, you can see the extent of the extramedullary disease in this patient was dramatic. Now, he was actually a very frail gentleman. He had been heavily pretreated. He had multiple issues. And so when we started on mezigdomide, frailty was a primary concern. As you can see from the bottom slide, his PET-CT was striking. And I can describe for you that actually clinically at the bedside, he had plasmacytoma growing across his porta and across his chest wall. After 4 months of mezigdomide dexamethasone, which he tolerated remarkably well with no hospitalizations, you can see the result, which was dramatic. My nursing staff were delighted because they could actually access his port, because his plasmacytoma literally melted away from it with therapy. So we were very struck by this. And this was a remarkable result. And the patient did well for a while. He subsequently progressed and then went on to other salvage therapies. But in a nutshell, this was one of the most striking examples to us of the activity of this oral therapy for extramedullary disease. Next slide, please. What about the pharmacodynamics? Well, as we published, what we were pleased to see, and in fact, the panel on the left is actually a patient of mine who was progressing on isatuximab pom-dex. And as you can see, at screening at bone marrow, his ALOS immunohistochemistry was strikingly positive. And after just a week of mezigdomide therapy, you can see the effect on the immunohistochemical staining with a follow-up bone marrow aspirate. And when we look more broadly across the study, you can see that this translated into effects on natural killer cells, T cells, and of course T cell subsets, which was particularly encouraging to see. And so we were also able to broadly look at the change in tumor ALOS as well. So I think one of the strengths of this study was and I want to especially acknowledge Celgene BMS because they really were phenomenal partners in this study, was the effort to look at these correlates and prove this effect. Next slide, please. What about combinations? Well, just a little early data here. This is from combination therapy with bortezomib, the response rate around 68%. Combined in the first cohort. Then in the second cohort, more striking at around 82%. And then similarly, in the combination with carfilzomib, we saw a solid signal around 62%. Again, no obvious difference between the PIs. Again, we were perhaps expecting carfilzomib to perform a little better in combination, but so far at least they've been very comparable. But again, we were particularly excited by the mezi-VD cohort there of 38 patients and an 82% response rate. So again, some interesting and provocative data with medium DORs for that particular cohort not reached and a manageable in profile. Again, infections are an issue, one has to be careful, but nonetheless, atypical, strange, or really severe or life-threatening infections were very uncommon. Next slide, please. This was to Sagar's point. The Excalibur study is ongoing, which I think is a very important trial. And then the successor programs are getting up and running, one focusing on bortezomib and one focusing on carfilzomib. In the successor 1 program, we're using the optimism regimen as the control group compared to mezi-VD. The slightly more controversial strategy with carfilzomib is a doublet versus the triplet, recognizing the power of KD. Thanos is actually the lead investigator on Successor 2, and I think this will be an important study, and we're certainly supporting it and will be part of it, but it's not the easiest one because of the control group, certainly in the US. Having said that, I think of all the doublets available, probably KD remains the one that I think we'll feel more comfortable with. But anyway, those are the phase 3s that are going forward. Next slide, please. And that's a quick whirly bird through the image and within the time frame. So welcome questions and discussion. Thank you very much, everyone.
Yeah, I think these are 2 really potent 3rd and 4th generation agents that I think we're excited to have. They both have some relative differences in terms of efficacy and adverse event profile, not dissimilar to the parallels between lenalidomide and pomalidomide, which I think are really important. But from my personal perspective, their best partners are the immune agents. They were engineered to be activators of the immune system, and I sort of describe them as an espresso for T cells. And I think that idea of being able to take an exhausted or non-responsive T cell phenotype and wake them up, we demonstrated that with the correlative work that Paul was referring to in patients who are getting the drug. So this is not just a preclinical model. This was a translational endpoint. And that to me really represents the idea of how do you make antibodies, T cell engagers, and CARs even better. And I think the use of these novel CELMoDs are great pathways to get there.
Yeah, sure. And I have a question, Paul, because here in Europe, sometimes the common question is how these new CELMoDs, iberdomide and mezigdomide, will replace the conventional ones. And indeed, there is a phase 3 clinical study in maintenance therapy. And Elena, I think that is the lead, comparing iberdomide with lenalidomide. How do you envision the incorporation of mezigdomide? Do you think that mezigdomide will replace pomalidomide, or, well, what is your input?
I think it's perhaps a little clearer for the IBA story in healthcare jurisdictions that will be able to, shoulder the resource challenge, because that I think is the biggest issue, is that, lenalidomide will remain a gold standard, especially in healthcare systems that, have significant cost constraints. I think the biggest advantage I see to iberdomide is this really important one, which is that the second cancer signal is likely to be much less, and so far appears to be minimal. There's strong scientific rationale for the difference. So second malignancies will be much less of an issue with IBRA, in my opinion, than they will be, or they are, with LEN. And so as we move this up front, for example, it's great to see the success of Elena's ISHIA study, because we're partnering IBA with isatuximab, bortezomib, dex in a quad. We're looking to develop IBA as an upfront induction remission strategy with IBA maintenance, so that we minimize the second cancer signal, and integrating that with transplant as well as with bispecifics, to Sagar's point. So we're looking at that. For the Mezi question, I think the potency is key. And I actually don't think it'll be one versus the other, Mary V. I really think we need all of them, to be honest with you. And I think as patients live 15, 20, 25 years, I want to be able to use everything, because, that is what I see day to day in clinic. I'd love to say I see drug vacations for 3 years. That's not what I'm seeing. And so I think having all of these pumps, hands to the pumps, is important. But I'm very struck by Mezi's activity in EMD, Mary V. Quite seriously, that's why I showed those two patient-focused cases, because Mezi, together with, we've obviously got melflufen, we've got isatuximab, we've got now Mezi, we've got certain drugs that are very good, and even for that matter, selinexor, that can be very good for EMD. So I think these are important tools to have. So Mezi perhaps for very challenging cases, in particular with EMD.
Yeah, I wanted, in fact, you to expand a little bit about this EMD, because we've heard the recent data from the CAR-T2 and the KARMA, and well, truly CAR-T alone, it seems that it's not the way to go with EMD patients. So probably combination of different strategies, that's the way for EMD, probably, where there is this high heterogeneity, so you have to really to hit different clones at the same time. So do you see something in combination? I don't know, Mezi plus what, how?
Yeah, no, I 100% agree, Elena. It's a great point. And I think EMD is not to be tackled with one agent, though. I really think it requires a rational combination approach. And we've got, so I think, very good drugs available to do that. And I really do think, because obviously, Elena, you and I were involved in the EMD project with melflufen. I mean, I think there's an opportunity for rationally combining, recognizing cytopenias are an issue, so you have to be super careful. But I do think we have a number of tools that we can combine to do this, and potentially on-ramp for things like CAR-Ts, because Hermann, maybe I'll speak to this more, but I know there's data from Germany now using, for example, melflufen to on-ramp because it doesn't penalize the T cell compartment into CAR T, and it's also highly immunogenic. You know, it upregulates calreticulin dramatically. So Sagar used the term, you need espresso for your T cells. We can rationally combine certain drugs that stimulate the T cells. So if we can navigate the kind of T cell exhaustion issue or that kind of space, and also manage the cytopenias, I could see cocktails in EMD utilizing a mezigdomide with other rational combination partners, and kind of using them as on-ramping strategies to cellular therapies or bispecifics and or both. I mean, that's ambitious, but I think not unreasonable to think about.